How an impulse moves from a neuron to a target cell. A target cell is what the neuron is trying to affect, like a neuron or a muscle or a gland. So here's one neuron, and it's sending information to this second neuron in this case. So this is the picture of an end of a neuron. And so you have a neuron here, and here's the axon of the neuron. It's kind of a bad picture. This is this neuron, and it is sending information to this second neuron. So in the previous videos, we were looking at this neuron sending information to this neuron. This time we're looking at this the previous videos were looking at how information flows along the neuron. Now what we're doing is saying how the information gets to from one neuron to another neuron or a muscle. And so that's what we're looking at here. Anything to do with this junction here where the neuron and the other neuron come close together is called a synapse. And so this would be called the synaptic knob, and these little vesicles in here would be called synaptic vesicles. And so this is the end of one neuron, and it contains synaptic vesicles, that's this, which contains something called neurotransmitter molecules. Long word, but it means what it says, neuro for neuron, and transmitter, meaning it's something that can transmit an electrical, or something that can transmit a nerve impulse from one neuron to another, or from one neuron to a muscle cell. And then the other really interesting thing about this is that the neuron doesn't actually touch the target. There's a space in between. And so there are some synapses where they actually are physically connected, but the vast majority of them are like this. So if this neuron fires, it doesn't necessarily make this one fire. In fact, sometimes it sends over inhibitory neurotransmitters, so the next one does not fire. But a lot of times they're excitatory neurotransmitters, which means this neuron is telling this one to fire. And whether or not this one will fire will depend on how many neurons are telling it to fire, how closely spaced the impulses are coming, and whether there's inhibitory neurons sending information to. So when a nerve impulse reaches the end of a neuron, we're going to kind of go through the steps of getting the impulse from that first neuron to the second neuron. So the nerve impulse has reached the end. Synaptic vesicles, that's these, fuse with the cell membrane and release neurotransmitters into the space by. So if you remember from our last unit, when you have a vesicle and the whole vesicle fuses and all the contents from that vesicle spill out, what do you call that? You call that exocytosis. So neurotransmitter molecules diffuse across the space. Neurotransmitter molecules reach these things. They're called receptor proteins. And those are receptor proteins on the target and bind to them. So if you look at this, here's a close-up of it. Here are all those cute little phospholipid molecules. And here's the receptor protein. And this is a very special receptor protein because it does two things. One is that here's the neurotransmitter. Maybe it's serotonin, maybe it's GABA, maybe it's dopamine or acetylcholine, whatever this neurotransmitter is. Once it fits in with the receptor actually, I'll show you a picture in a minute, but the receptor can change shape and allow sodium in. If sodium's coming in like this, hopefully that rings a bell from our last video. When the sodium comes in, that starts a new nerve impulse. Cool, huh? So those are the receptors. All of these purple things are the receptors. So when the neurotransmitter binds to the receptor, oops, it hasn't bound here, but yep, it has bound here. So when the neurotransmitter binds to the receptor on the target, the receptor protein changes shape and allows, so nothing can go through here. Now all of a sudden this is open, now things can go through, and it's actually allowing ions to move through, maybe starting a new impulse. So remember, ions are the electricity of the body. Once the sodium moves in and the potassium moves out, that might start a new nerve impulse in the next neuron. Whoa. What process is used when a synaptic vesicle releases neurotransmitter molecules into the space between the neuron and the target? And that is exocytosis. If there are 50 different neurotransmitters, then how many different types of neurotransmitter receptor molecules should exist? There should be 50 different ones because they're all specific. So serotonin is a neurotransmitter. There are receptors for serotonin. Those receptors are totally different than the receptors for acetylcholine. All right. Stopping a neurotransmitter. So you want to have a lot of control over when the target is stimulated. So for example, if your neuron stimulates your muscle to contract and then your muscle can never relax, that's a really bad thing. So you want to have very fine control over when the neurotransmitters are exciting another neuron or a muscle or whatever. So there are three things to know here. A neurotransmitter bound to a target continues. Sorry, there are three ways to stop a neurotransmitter, but let's talk about why it's important first. A neurotransmitter bound to a target continues to affect the target. This should not occur for long. It's important to have control over when a target is stimulated. And again, that's like making your muscle contract and then never letting it relax. You definitely want to let it relax eventually. And so here's a picture of that. You've got your first neuron, and let's say this is a second neuron. This is the vesicle fusing for exocytosis. It's letting the neurotransmitters out. In this case, the neurotransmitter is acetylcholine. Here's the acetylcholine binding to the receptor, and when that happens, sodium can move through, and you start a muscle contracting, or you start a nerve impulse, or whatever. Take a look at this thing. This thing is called acetylcholine. This thing is called acetylcholinesterase. Hopefully you remember that ACE means it's an enzyme. And look, it's taking the acetylcholine and it's breaking it down. So as soon as the acetylcholine is released, whatever bounces into here gets broken down. So that's one way that you can get rid of neurotransmitters. So removal of a neurotransmitter occurs three ways. One is enzymes breaking down the neurotransmitters. Another is that the neurotransmitters can be taken back up again, so reabsorbed by helper cells or by the first neurons. So here's a neurotransmitter that got released, and they're going to bounce everywhere because it's diffusion, right? Brownian movement, they just bounce, 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 bounce. Some of them will bounce back up here and get reabsorbed by the first neuron, or there might be a helper, they're called glial cell, and maybe that one will bounce, 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 and bounce into this one and get taken up by this one. So it's called reabsorption. And then finally, diffusion away from receptors. So they'll just kind of diffuse away. Goodbye. Okay, cholinesterase is an enzyme that breaks down acetylcholine. Acetylcholine is a neurotransmitter that's responsible for many things, but one of them is to stimulate muscles to contract. How could a cholinesterase inhibitor be used as an ant poison? And actually there are ant poisons made with cholinesterase inhibitors. And so you can think about that and pause it, and I'm just going to tell you now. A cholinesterase inhibitor, that means you're going to stop cholinesterase from working, which means the junction between a neuron and a muscle cell is going to have way more acetylcholine in there than it should, which means that the muscle will be contracted way longer than it should. And so that's going to interfere with the everything about the ant's skeletal muscular system, and the, beating heart, all that stuff is going to not work. And so they're going to contract too much, and that's what actually will kill the ant. Okay, human brain functions. Let's do this. This won't take too long. So what I don't need you to memorize any of this. What I want you to know is that the brain is specialized and different areas do different things. So this is the back of the brain, and this area is the primary visual cortex. That means this is the part responsible for you being able to see. So I'm looking out my window now, and I see a pretty yellow house across the street from me with some pine trees, and I see this green recycling bin. This part is called the visual association area. So this part is what allows me to see the yellow house and the pine trees. This part is the part that lets me say, oh, you know what? My dad lives in that house with my sister and my nephews, and it's a great place, and I, can picture all the different parts of the house, all that stuff. And that recycling bin, I'm looking up there thinking, yep, I got to take that in pretty soon too. So there are different functions for different parts of the brain. Let's see. If you look over here, this is the auditory cortex, so that's for hearing. The prefrontal cortex and the frontal lobe, this part's really important for problem solving and for thinking, for judgment, all that stuff. This part's really actually kind of interesting. This is the primary motor cortex, and this is the primary sensory cortex. And so this part is what makes you have control over skeletal movements, and it's cool because it's upside down and backwards. So if this is the left side of the brain, this would control your right foot all the way up to your right face. And if this is this part, if this is the left side of the brain, this would be the one that would say, ooh, did I say left? Yeah, if this is the left side of the brain, then it would say, my right foot hurts. or my right elbow hurts, or I have an itch on my nose, that kind of thing. So they're both backwards and upside down, which is, I think it's kind of cool. Human wiring is kind of fun. So the brain can be understood two ways. One is chemically, due to neurotransmitters. And so there are various drugs that have been produced to affect neurotransmitter levels, such as SSRIs. So SSRIs include things like Zoloft and Paxil, and they're kind of cool because the way that they work, here's a neuron telling another neuron what to do. Serotonin, selective, this one's for serotonin. Serotonin is the neurotransmitter that you need to feel any degree of happiness. So people who are depressed tend to have less serotonin around than most everybody else. And so your serotonin's released, and let's say this is not very much. Let's say you're depressed and not very much serotonin is getting through. Well, this is a selective serotonin, meaning it works for serotonin and not dopamine or epinephrine or whatever. And this means reuptake inhibitor. So that means the serotonin should be taken back up by this neuron, but this drug is stopping it from being taken up. That means it's going to stay in here longer, and you'll have more serotonin in your synaptic clefts to affect the next neuron. And so your brain is going to function more like a normal person who doesn't have depression at the time, or maybe like your brain does when you're not depressed. So those are kind of cool drugs. I think it's interesting that, I think it's changed, these drugs have changed the way people have really thought about some mental illness. And if you have diabetes, you would never think of not having insulin. And so I think when I was a kid, people, didn't talk about mental illness, and now people do because it's seen as a medical issue. Okay, so the brain can also be understood electrically, and this has to do with the ions that move during nerve impulses. And so um, EEGs can be used to focus on electrical signals, and I think I have a picture of it. So these are some of the electrical signals. This is alpha, beta, theta, delta, and it's actually in messed up order. This is the 4 hertz or less. This is very slow waves. Faster, faster, and this one's the fastest. I'm sorry I have these sort of in the wrong order. But anyway, this is showing you really fast waves, and this is showing you really slow waves. If you look at this, that's actually considered a wave. And the brain should have different brain waves at different times. So for example, if you're awake and reading, and you're looking at the lobe in the back there where vision is, you should have a certain wavelength that would be different than if your eyes were closed. And so there are, various types of research looking at brain waves, which is kind of cool. And that's it for today.